Here we go. Down in my reptile room, and I want to talk to you guys a little bit about corn snake genetics, since we're studying genetics in class right now. All right, so you might remember these guys from class. We've got Melanie, the amelanistic corn. She does not make the black pigment. All right, she does not make black pigment. She does make the red and orange. Now, one thing that makes her a little bit different is she also doesn't have the checkerboard belly. A lot of snakes that corn snakes have that checkered belly. It's one of the characteristics of a corn. But we talked about how things are a little more complicated than the cut and dry dominant recessive stuff. So this is Melanie. She is known as what's known as an amelanistic corn snake, or incomplete albino. She doesn't make the black. She makes the red and orange. She is homozygous recessive for the melanin gene, the gene that makes the dark pigment. She's little m, little m. Now, as far as making red, I don't know if she's heterozygous or homozygous dominant for the red. You would have to either check her DNA or do some test cross to determine. All right, here over on the right of the screen, I'm showing you what a normal corn snake would look like. It does have the red and the orange, but it's got the well-defined black in there as well. None of the corn snakes that I have as pets are normal corn snakes. So let's look at what I was saying about Melanie, whether we don't know if she's homozygous dominant or heterozygous for making her red. So what we do know is that, remember, we're following two genes here. This is a dihybrid cross. And again, Melanie is what's known as an amelanistic corn snake. Melanin is the pigment that makes skin dark in snakes and in humans. And she is amelanistic. She does not make that melanin. So we know that for the melanin, she's little m, little n. All right, two little m's. Now, for making the red pigment, we're just going to use R for the red. What we don't know is if she is big R, little m, or she could be big R, little m, because both of those genotypes would result in the same phenotype, which is what she is, an amelanistic corn snake. All right, let's move along with the video. Determine that. So this was Melanie from our class. She says hello. In this container, we have Tiffany, which is the biggest snake I have, one of the oldest right now. She is not wanting to come out of this bamboo right now. She is aneutheristic. She makes the black pigment, but does not make the red. She's homozygous recessive for making red. Now, her belly has that checkered pattern that most corn snakes have. In the wild, if you were to find a corn snake, they would most likely have this checkered belly. They would have this same pattern on the skin, but they would have the red in the pattern like Melanie does, and they would also have the black in the pattern like Tiffany does. All right, maybe she'll come out here in a second. We'll play with her. And here she goes. So here is Tiffany. Come on out, girl. All right. Now, I don't know, or I think I know now, but I want to tell you the story behind why I know that. That she is most likely homozygous dominant for her black, meaning she's big M instead of big M, little m. And I'm going to tell you how I know that in just a second. It has to do with the fact that she has babies, and I'm going to show you one of her babies. Here she comes. So here is all of Tiffany. All right, there we go. There's Tiffany. She's a big girl for a corn snake. All right, I'm gonna put her down right here. All right, now we have William Snakespeare. He is about the same age as Tiffany, but he does not eat as much. He is also a anutheristic corn. All right, anutheristic corn snake. And again, I believe for that making black, for the melanin gene, I believe he is homozygous dominant as well. At least one of these two is homozygous dominant, and I'm going to tell you why I know that. Now, you'll notice that there's something else going on with William. He doesn't have the same pattern that Tiffany does. He's got a more splotchy pattern rather than some of them kind of look like ghosts going down in that kind of U circular shaped pattern going down. He's got a bunch of circles, and it's just a different pattern. I don't know the best way to describe it. His belly is plain, it's not checkered. And he's got a stripe going down most of his body. This is something else called motley that we're going to discuss in just a minute. Now, like I said before, I've got Tiffany and I've got William Snakespeare. They are both corn snakes. All right, let me try to hold them back up together. All right, now Tiffany, readjust this lighting. They are both anutheristic corn snakes. They don't make the red pigment.
All right, so again, let's go in and investigate this a little further. Now, I keep calling these by the names we named them in class: Tiffany, William Snakespeare, Oreo, Melanie. But don't forget, these things have scientific names. These are all corn snakes. The scientific name of a corn snake is a Pantherophis guttatus. So we cannot forget how scientific names work. Pantherophis and then guttatus. All right, so we have been studying science, so we do need to remember these things have scientific names. They're in the genus Pantherophis, and the species name is a guttatus. All right, so do not forget that. Now, these particular snakes, pardon me, these particular snakes. So as I was saying, these particular snakes, these are Pantherophis guttatus. Another name, another common name for a corn snake is a red rat snake. This genus Pantherophis is actually the same genus that a black rat snake is in. They're much more common. You've probably come across a black rat snake before. Those are Pantherophis or Pantherophis obsoletus. These are Pantherophis guttatuses. So they're in the same genus. The corn snake is also known as a red rat snake. Now, these particular corn snakes I have are called anutheristic. And I'm going to spell that out for you because that is a little bit of an unusual word. Anutheristic. Anutheristic. And another word for this is black albino. Sometimes it's called a black albino because it's albino in the sense of it doesn't make the red, it does make the black. So usually corn snakes have black and red. These don't make the red. So if you're looking at the genes of these guys, and we're following the gene for making red and the gene for making black, we know their genotype for making red is little r, little r. What we don't know is if, by looking at them, you can't tell if their genotype for making black, and we're going to use M for the black, for melanin, they could be big M, little r, or they could be big M, little r, little r. This has implications if you're trying to breed them and get something that has this, little m, little r, like our nose. That's known as a snow corn, which we're going to talk about in a few minutes. Because in order to get a snow corn, from something that looks aneuploristic, you would have to have two parents that are heterozygous. If either parent is homozygous dominant for the black pigment, then there's not a chance of the baby getting two little limbs. And we're going to look at that in just a minute. Let me play just a little more of this.
And the reason that I am saying that at least one of them is homozygous dominant for making black is because they've had babies. All right. They had, I think it was 17 babies. And because of the results, I got half. I'm sorry, I was thinking ahead of myself. Because of the results of their babies, I can tell you that it's most likely at least one of them is homozygous dominant for the black pigment. And that's because if they were both heterozygous for the black pigment, which is what I was really hoping, if they were heterozygous for that pigment, then I should have gotten 25% of the time I should have gotten a snow corn, and I was really hoping for a snow corn, but I did not. I think there was 17 or 18 babies. I don't remember the exact number, and every single one of them was aneuploristic. In fact, you've seen a couple of the babies. One of them we had in class. It's over here. It's the one that we ended up calling Oreo. Oreo is the baby of Tiffany and Melanie, and you can see that it makes the black but not red. But it's got that pattern more like the dad, all right? It's got that pattern where it's got more lines going down its body. It doesn't have the splotchiness, ghosty pattern, and it's got a plain belly, Oreo does. So that's something else I'm going to talk about here in just a minute. This is another one that you haven't met. This one stays in my son's room. This one we call Ghosty. All right, this is a female, and it takes after his mom. She takes after her mom. The reason I call it Ghosty is if you look at the pattern on its back, there's a couple of those splotches that look like little ghosts. There's one there. I think there's like a couple of them. That might have been the most prominent one. There's a couple of the ghost marks on it. That's just something I call it. That's not an actual thing with these guys. I just like this one because it had a real distinct, there it is again, the ghost-like shape on it. Let me get a good picture of the ghost. There we go. Now, so because when these two mated, every one of their babies came out making the black pigment. That kind of lets me know that they're not carriers for the gene that is defective for making melanin, because if it was, chances are there would have been a snow corn. This still could have been just by coincidence that there was no snow corns, but Oreo and Ghosty have actually made it again, and some of you might have seen at the beginning of the year, not the beginning of the semester, they had babies. I ended up getting rid of their babies, I did let a few of them go. Because I couldn't take care of more snakes, and all of their babies came out making black as well. So at least one of them is homozygous dominant for making black as well, and we can look at a Punnett square and see that. The other thing I want to talk to you guys about.
All right, so here is a picture of snow corn, which is what I was really hoping we would end up with. Now, in order to get the snow corn, you would have to have a snake that is homozygous recessive for the red and for the black. Now, we know with our aneuploristic ones that they're homozygous recessive for red. So we know that they're both this. All right, so both parents are going to be little r if we cross them with each other. Now, in order to have a snow corn, we have to have each one be heterozygous. Now, before they mated, I did not know whether they were big M, little m, or big M, big M. Now, we could sit here and do a dihybrid cross, but since we know that for red, all there are little r's, it's kind of pointless to set up a Punnett square just to check the little r's, because they're all going to be little r. So let's just look at the M's. That's all we really need to do for this. So I have these two corn snakes that mated, and while I was waiting on the eggs to incubate, I made an incubator, and it took a couple months. They actually hatched during school, which was really cool, around 1 October. What I didn't know was if the parents were big M, or big M, little m. Now let's say that there was one of each. So if one parent was big M, little m, and the other was big M, and you do the Punnett square, this is what you end up with. They're all going to get at least one big M because that one parent's always passing on the big M. So if either one of them is homozygous dominant, I'm not getting any snow corns. All right. The only way to get snow corns is if both of them were heterozygous for their melanin. And if that's the case, that they were both heterozygous for their melanin, we get that Mendelian, that Gregor Mendel 3-to-1 ratio with his pea plants, where you would get a 75% chance of the babies looking black and a 25% chance of the two little M's coming together. However, I had about 17 babies and none of them came out snow corn. And then 2 of the babies made it again a few years later and none of them came out snow corn, which lets me know that at least one of these 2 aneuphoristic snakes is homozygous dominant for its melanin. It could be both of them are homozygous dominant, or it could be just one. There's no way to tell without doing some other stuff. Now, kind of interesting here, I did go back and find some pictures of the babies. These are the eggs. These are the eggs that one of these babies is Oreo and one of them is Ghosty. This is from about 4 or 5 years ago, maybe 4 years ago. This is Tiffany when she had just laid the eggs and was still kind of wrapped around them and emaciated. And then I moved them out of there so they would incubate because they won't incubate well in her container. And I made an incubator for those eggs and they hatched. I think all but like 1 or 2 of the eggs hatched. Here's some of the baby snakes we were passing around in class. They're very cute, nice big eyes, little head, very cute little aneuphoristic corn snakes. And you can look, let's see if we can see the different patterns. About half of them came out with the normal pattern. I think this is one with the motley pattern. Some of them developed some yellow later on, but when they were first hatched, they didn't have it. Their patterns do change as they get older a little bit. There's one with the motley pattern. I think this was one with the regular pattern. And it was about half and half, which we're going to talk about with the motley stuff. But out of all of these, none of them came out snow corns. So here's pictures of those babies. It actually takes them a couple days to fully come out of their shell. They go in and out of their shell for a few days. It was very exciting times. And then this all happened again last summer, the beginning of this year, when 2 of these babies actually mated and had more babies. So that is the whole thing about the snow corn. Let's move into talking about the motley pattern.
And see that. The other thing I want to talk to you guys about is this other pattern. The fact that this one over here, William Snakespeare, the fact that he has this unusual, this different pattern where he's got stripes down his body and he's got a lack of checkers on his belly. All right. So another thing we could talk about is this. This is called the motley pattern. The different pattern and the lack of belly spots, or the checkered belly. The lack of that is what's called motley. And what goes along with motley a lot of times is the stripe, which is another one called striped, but his stripe doesn't go all the way down. His stripe is only partly down his body. So the striped gene is linked with the motley gene, and it's a little more complicated. Like we talked about, a lot of these cases are more complicated, but the motley itself is a recessive pattern, and that I do have some proof of.
So with the motley, that is a clear cut, at least just the plain motley. Now the stripe is a little something extra to it. But with the motley, it follows more of a regular Mendelian inheritance pattern. So let me write out what this is. This is called the motley. So this one is an aneuphoristic and motley. There's many genes that make up a snake, just like there's many genes that make up of you or me. So this is an aneuploristic and motley, and we've already been using M for the melanin, so let's use T to represent a normal, meaning the normal pattern, and lowercase t to represent the motley. And this is a simple dominant recessive thing. So looking at William here, this Pantherophis guttatus, the aneuploristic motley corn snake, because he's motley, I actually know something about his genotype. It is little t, little t. Now, if I'm looking at a snake that is not motley, all right, like Tiffany, the one that has the regular pattern, she doesn't have the stripe. She doesn't have the kind of lines going down the side and the circles like that. That's what defines a motley, or the non-checkered belly. So she is either big T, or she is big T, little t. Now, if she were big T, we could do a Punnett square, and we could see, I'm going to go to the other side here, we could see that if Tiffany was big T, then none of her babies should come out motley. They would all have that one big T. But that's not what happened. What actually happened was I had 17 or 18 babies, and I had about 9 one way and 8 the other way. I was just showing you those when they were coming out of the egg. One of them is Oreo and the other one is Ghosty. So the babies, what I ended up getting was exactly what you would expect if Tiffany was heterozygous for her pattern. That would mean that there's a 50% chance that her babies are going to come out motley, because little t equals motley, and that's the only way to get motley. So that tells me, because of looking at the genetics, that Tiffany, not the snake in the picture, but the other one, the real big one, the fatter one, she is heterozygous for her pattern. So I know something about Tiffany. She's heterozygous for her normal pattern. She's a carrier of the motley gene. And the one you're looking at here is a motley, which is homozygous recessive. So he is here, the 2 little t's down in the bottom left, and Tiffany is the heterozygote. And if they mate, their babies should be half and half. And the babies that look normal, like Ghosty, who I showed you, is actually a carrier for motley, which means if he mates, he's going to pass it on again. And in fact, he mated with Oreo, and their babies came out, about 50% of them were motley and 50% of them were not. So it's an example of a Mendelian trait. Well, let's finish up the video here.
That I do have some proof of, because like I said, Tiffany and William have mated. Tiffany is going back into hiding. She doesn't want to be out right now. She is under her rock there. So Tiffany is not motley. So she's got the regular pattern, and then we have this motley. Now, when they had babies, all right, when they mated, it was an accident that they mated. I didn't mean for them to mate. It happened, which is why they're in separate cages now. I didn't know if she is homozygous dominant for her pattern or heterozygous. It turns out she's got to be heterozygous, because she mated with William here, and William is motley, which means homozygous. He's homozygous recessive. In order to show motley, he's got to be homozygous recessive. Now, Tiffany could be homozygous dominant or heterozygous. The thing is, they had about 17 or 18 babies. I'm thinking now it was like 17, and 8 of them came out motley like the father. And 9 of them, well, I don't remember the exact numbers, but it was half and half. About 8 of them, 7 or 8, came out like the father, like this, showing the motley, and about 7 or 8 of them, whatever the numbers were, came out like Ghosty, who had the regular pattern like the mother. Now that tells me that the mother, Tiffany, must be heterozygous for the pattern. Otherwise, the babies would not have come out motley. If she was homozygous dominant, all the babies would have been all the babies would have been the regular pattern. Also, Oreo and Ghosty mated, and their babies were also about half and half motley and regular pattern, which shows that also Ghosty here, Ghosty would have to be heterozygous for his pattern. Now again, all of these are homozygous recessive for
Good example of the ghost, which gives him his name right there.
making the red. All these black and brown ones I'm showing you, they're all homozygous recessive for making red. The ones that have the motley pattern are homozygous recessive for the motley gene. And the ones like Ghosty and Ghosty's mom have to be heterozygous. They couldn't be homozygous dominant because if they were, their babies would not have come out with motley. The only way for that to have happened is if they were heterozygous. And if they're heterozygous, you can look at the Punnett square and you can see that a Punnett square between a regular pattern right there and between, a Punnett square between a regular pattern and a motley. If this is a heterozygous for the regular pattern, the Punnett square should reveal a 50.
There we go. So right there in front of you, you can see the two snakes, and we can actually put this into action and look at a very clear-cut example of Mendelian simple dominant recessive. Because over here we've got, remember they're both aneuploristic, but over here we got the aneuploristic normal pattern snake, which is either big T, or big T, little t. And over in the left corner here, we have the motley snake, which has to be homozygous recessive if this is truly a dominant recessive thing. And they mated, and when they mated, I got a 50-50 mix of the babies, where 50% of the babies came out to be motley, and 50% of the babies came out to be normal pattern. And the only way that this could have occurred would have been if obviously the motley one is little t, and the one with the normal pattern would have to be big T, little t in order to make this work. That's the only way this would have worked out in this case. So that tells me that this snake here is not homozygous dominant. Ghosty must be heterozygous, just like her mother is.
50 mix of motley and regular in it, and that is what I ended up seeing in 2 generations. Now he just jumped out of my hand and onto the floor. So there is a little snake genetics for you, brought to me from my reptile room here during the coronavirus isolation of 2020 distance learning for my biology classes. I hope this video helps shed some light on some stuff we're talking about, and that it finds you well, and I will be speaking with you again soon. Take care.
And take care for me as well.